is talk about some topics related to caries. There's a lot of material, and I've got about less than 2 hours to deal with it, so I'm going to be selective. I'm going to focus a little bit on epidemiology so that you get an understanding of the severity of the disease nationwide. I'm going to focus on the early, just a little bit, on the early papers that identified bacteria and that identified strep mutans, as well as papers that identified secretory IgA. Now, you know about secretory IgA a little bit? Has anyone spoken to you? Did Dr. Levine talk to you about secretory IgA as part of the antibody complex, but maybe not in the context of the oral cavity? So we're going to talk about secretory IgA in the context of the oral cavity, because it is extremely important as a molecule that helps us get rid of excess bacteria in the oral cavity. At the same time, in the context of caries, there's a lot of work going into identifying if one can utilize, by hyperimmunizing the host, if one can utilize specific secretory IgA antibodies that home in on specific virulence factors of cariogenic bacteria. So we're going to focus on secretory IgA. And obviously we're going to focus on the cariogenic bacteria. And we're going to talk about certain virulence factors that are being studied today in regards to that process. So you'll hear me talk about adhesin molecules that bind to anchoring molecules of the salivary pellicle. You'll hear me talk about glucosyltransferase and maybe fructosyltransferase, which are enzymes that promote glucan development. We'll talk about glucans. Glucans are these extracellular polysaccharides that surround the body of the organism to make it sticky. Because one of the problems with strep mutans for itself, the problem is that it doesn't bind well to salivary pellicle. So it's got to have a strategy that outwits this, that overcomes this, right? So there are a lot of bacteria that evolved over the millennia to bind specifically to salivary pellicle at very high affinity rates. And we'll talk a little bit about that. So those certain commensal bacteria that have evolved to bind to the tooth readily, that's a good thing. That's what you want to happen. That protects us from the likes of strep mutans. But all you need is one bug to be able to bind to these salivary pellicle anchoring molecules to get the process rolling. So that if the subject, the host, starts eating sucrose, then that one bug has a way to circumvent its inability to bind to salivary pellicle by promoting the production of these glucan molecules where other strep mutans can bind to. So we'll talk about glucans. We'll talk about a really interesting topic that you as clinicians-to-be need to convey to the parent of the child, because the parent may come to you and say, oh, I've completely stopped giving my child sucrose. No sucrose, zero sucrose. And yet 6 months after you've done extensive reconstruction in the mouth of a child with multiple caries, 6 months later, there are new caries. Now why is that? If the parent is telling the truth, but indeed the child has no sucrose in those 6 months. There's a reason for it, and I'll explain why in terms of strep mutan strategies to overcome that. So the take-home message is it's not enough to stop with the sucrose. You've got to get rid of the excess strep mutans. Importantly, I won't have time to go and discuss anything from slide 34 on. All right? So I'm not going to hold you responsible for anything from slide 34 on. It has a lot to do with, I won't call it science fiction, but some new stuff, new old stuff related to molecular biology and how certain antibodies are being produced as ways to combat the virulence of strep mutans. I will focus a little bit on what's called hyperimmunization. The whole topic of using science to combat the virulence of an organism that is not life-threatening is in question. Think about it. If you've got a life-threatening pathogen, you're going to do everything you can to get rid of it. So that justifies maybe producing a vaccine. But if you've got an organism like strep mutans, which is responsible to a great degree, over 80% of caries is associated with this bug, does it make sense to create a vaccine against this kind of organism? I contend it doesn't make sense, because there is morbidity, or some level of morbidity involved in mass vaccinating people. And what is today's solution to caries, aside from the dentist? What is it? Huh? Yes. All you need to do is make certain that the fluoride levels are 1 part per million, and you've got coverage. Especially when it's a baby, where the deciduous teeth are evolving in the jaws, and subsequent to that, when the permanent teeth are developing in the jaws. So eradication of caries can happen in a way that doesn't require vaccination, right? So the FDA is not going to permit a vaccine to be mass used with a disease that isn't life-threatening overall. So that's an important message. Okay, so with that as an introduction, this is mass caries. Okay? Tremendous caries in a child. The problem is, this is the wrong slide. I wanted to find a slide as quickly as I could that demonstrate mass caries due to sucrose. But this is not mass caries due to sucrose. This is a better slide for innate immunity. Read what it says. In a patient with impaired salivary function as a result of radiation therapy. So that is an example. If you radiate the salivary glands, you're going to have dry mouth, you're going to have a reduction in a lot of the innate response elements that we talked about in the last 2 presentations. This is a take-home message. But it works well also for caries related to strep mutans, because you're going to see things like this in children, just as you do in the unfortunate situation of a child that may have cancer that requires some kind of radiation therapy. So you're going to have to treat those kinds of situations. Caries is the outcome of immune deficiency, as well as, in the case of healthy children, overuse of sucrose. Okay, so just keep that in mind. All right, what are the learning objectives? The importance of secretory IgA. That's going to be a take-home message. I'm going to just barely touch on what's referred to as naturally induced immunity versus artificially induced hyperimmunization. There is a place for injecting someone with a particular antigen that's been identified as being virulent in an organism. There is justification. And what you get is hyperimmunization. Anyone got the flu shot before the winter started? Okay, that's hyperimmunization. That's in order to reduce your chances of getting influenza. Now that's life-threatening to either babies or old folks. So the question still remains, is there justification for doing hyperimmunization in a non-life-threatening disease? Now there are the exceptions to the rule, where these caries situations are left untreated, you get abscesses, and there are these rare cases where these abscesses can travel up arteries to the brain and create havoc in the brain. Very, very rare, but does that really justify hyperimmunizing mankind? What are the criteria for karyogenicity? How do you call an organism a bacterium karyogenic? What are the criteria for calling them that? The family, the umbrella family of mutans streptococci, in which strep mutans is one of the members. Strep mutans, virulence factors, and properties for adherence. We're going to talk about some of those virulence factors. The Embden-Meyerhoff pathway, I'll just mention it. It is the pathway by which glucose, which is the monomer from the dimer fructose glucose that makes up sucrose, is metabolized so that at the end of the day you get lactic acid. An acid that isn't volatile, it stays around a lot, and it promotes the degradation and the destruction of the enamel. Keep in mind, we're not talking about lactobacilli. It's important to mention that the secondary organisms that come into play to promote more destruction of the tooth are the lactobacilli. And lactobacilli overall are good bugs in the mouth. But they require organisms like strep mutans to start the process of enamel destruction so that they can get in and start destroying the dentin. All right? Lactobacilli are not good at destroying the enamel. Organisms like strep mutans are. And then, I'm not going to be talking about this. All right? That's from 34 on. We'll talk about the epidemiology, caries and secretory IgA, mutant streptococci, in general, strep mutans. A little bit about designing an anti-caries vaccine. What do you need in order to design a vaccine? What is the first thing that you need in order to design a vaccine? What do you need? You need a pathogen. You need an organism that you've identified as being the culprit of the disease. What's the next thing you need once you've identified the organism? Huh? Right, an antigen. A virulence factor that the bug produces that you then call an antigen because it's foreign and goes into the body, generates antibodies. So that's an antigen, exactly. So you need to identify the virulence factors. That's why I'm going to introduce the virulence factors to you, but I'm not going to go into detail about generating the vaccine. And this part I'm not going to be talking about. Okay. A little bit about epidemiology. The busy slide. What do you need to know? So back in 2000, the U.S. Surgeon General's report, generated by David Satcher. David Satcher is an MD who graduated from Case Medical School, became the Surgeon General then, ordered a report to be conducted on the epidemiology of caries. They call it a silent epidemic. And obviously, poor children, elderly, members of racial and ethnic minority groups are at high risk for this. Okay? Now, this is important. About, I would say, fluoridation, which we talked about earlier, reduces caries, or reduced caries up until today, of about half of the children in this age group. But the CDC estimates that greater than 80 million people in the United States are without fluoridated water. Does anyone have a reason, a suggestion as to why that may be? Some of it's political. Yes?
The use of flowing water in places that don't have.
Right. But why would people object to having fluoride in their water? We have a constitution. Yes?
Is it just like one more thing that the government is doing without the...
Yeah, something like that. We have a constitution, the First Amendment. There's nothing like it in the world. And sometimes that's a good thing, but sometimes people take it to the extreme. I don't know why, but there are people who believe that fluoride is poison. Just like there are certain people who believe that vaccinating their children is a bad thing overall. And then there are repercussions from that. So there are states where referendums go up and down about fluoridating water. And they talk about, well, there are rat studies that indicate that if you put fluoride in their food, they'll develop tumors. But how much fluoride do they? They pump them up with fluoride to levels that would kill a human, right? And there are things like fluorosis that you may be studying, where there's excess fluoride in the water, in well water, for example, that communities drink from. And the babies that are born in those communities develop these striations in their enamel. Sometimes they're brown spots, right? So there is discoloration that occurs, but not at 1 part per million. And I would hope that in community dentistry they're teaching you about the importance of the right concentration of fluoride in the drinking water. And where there isn't fluoride, there are pills. I was a postdoc trainee at the University of Texas in San Antonio in 1988-89. I had 2 little babies. There was no water in this, and no fluoride in the water in San Antonio. It was around the time that a state referendum was being voted on about fluoride in the water. But the Texans, they're unique. They don't want to have anything put in their drinking water. 0. Now, they don't filter their water, so they don't know what they're drinking sometimes. But they don't want fluoride in the water. Well, I had no choice but to get fluoride tablets, 1 ppm. My kids today, Dove is 32, Tali is 29, 0 caries. Never. They've never seen a pediatric dentist. They got the most beautiful teeth on the planet, all because of using these little pills that you can get very cheaply at the pharmacy. So you can overcome the First Amendment. All right? Anyway, all of this is just to impress upon you the need for action. All right? This is how much was spent in 2010 in dental services in the United States. You guys have a future. Okay. So the National Anti-Caries Strategy that came out from this big study suggested that we need to combat the microbial agent. That's why NIH and other agencies that are doing research at the time were promoting the idea. Program announcements came out for researchers to submit grants to generate vaccines. That was in the year 2000, in response to the Surgeon General's report. And so a lot of individuals, colleagues of mine, the University of Florida, Pennsylvania, some others, came out with whole notions of vaccine candidates. Why? Because they were working on identifying the virulence factors of cariogenic bacteria. So to combat the microbial agent, to increase tooth resistance, well, obviously fluoride does that. Obviously, to modify the diet, you have to educate the parent. in order to reduce sugar intake in the kids and to deliver anti-caries measures to the public. So these were, in response to the U.S. Surgeon's report, these were directives coming out of the government to try and promote these kinds of activities. The American Dental Association got involved, and I don't know to what degree, almost 20 years later, we've really made a dent in the caries epidemic. A little bit of history. This guy in 1924, in this journal, British Journal of Experimental Pathology, discovered and isolated strep mutans. All right? And he reported on it. And no one believed him. 1924. I don't know what they thought caries, what the reason for caries was, but they didn't think it was this. In 1946, in the Journal of Dental Research, these guys did some experiments where they fed rats with and without penicillin this guy, Lactobacillus acidophilus, to show that dental caries were associated with Lactobacillus acidophilus. Now, Lactobacillus acidophilus is a lactobug that can cause exacerbation of existing caries. What that showed was that just about any, well, the take-home was that just about any gram-positive fermenting organism, if you put a lot of it into a rat's mouth, okay, and feed it the kind of chow that will generate caries, and you don't give penicillin, you're going to get caries. All right? And if you do give penicillin, then you're not going to get caries. This was the first real in vivo experiment that demonstrated that fermenting organisms were involved in the production of caries. And these guys in '54 in the Journal of Dental Research used gnotobiotic rats. Do you know what gnotobiotic means? Meaning grown in a sterile environment. They don't have bacteria at all. But what they did is they fed them high cariogenic diets. And the cariogenic diet was sterile. Just had a lot of sugar. That alone is not enough to cause caries. All right? So these are stepping stones of findings that indicated that, most importantly, you need bacteria, you need the right kind of bacteria, and you need substrate, and that is sugar. You put those things together, and you'll get caries. And if you treat them with an antibiotic, you'll reduce the incidence of caries. And then in the mid-60s, after a number of epidemiological and etiological studies, this guy reemerged as the causative agent, to which we now believe plays a role in about 80% of caries, at least in the United States. All right, now there's work coming out of Ohio State. Ann Griffin and her husband Gene Lace is a microbiologist. She's a pediatric dentist, well known in the field, and he's a microbiologist. They've been doing microbiome studies in babies and young children. They've identified other cariogenic organisms in the carious lesions. So the verdict still isn't in as to whether this organism is exclusive in doing that sort of stuff. There's another organism called Strep sabrinus that we're going to talk about that makes up maybe 15 to 20% of cariogenic in the etiology of caries. So my belief is that this is not the only organism. My belief is that this organism plays a major role, but clearly is not the only organism. Now this guy Tomasi in '65 all of a sudden introduces a host response molecule called IgA. Okay? So now all of a sudden there's something that the host is generating in response to caries. And that's the immunoglobulin A. So what do I say here? Dental vaccination approaches targeting a specific pathogen and manipulating a specific humoral immune system. Okay, and that's what the crux of the presentation today is about. So what about secretory IgA? What do you need to know? What are things that are important in regards to secretory IgA? First of all, at birth, there is no secretory IgA in saliva. Zero. And what's interesting is that the organisms that make up the first wave of streptococcal organisms that colonize the baby's mouth are organisms that are not karyogenic at all. These are important organisms that bind to mucosa, soft tissue. Streptococcus mitis, Streptococcus salivarius. And there are no antibodies to strep mutans. Zero. Why do you think that is? And this is at this period of time, between 16 and 28 weeks. Divide that by 4, that's what? That's 7 months? 4 into 20, no, 4 into, right? So somewhere up to 7 months of life, there are still no antibodies to strep mutans. Why do you think that is?
So those are good bugs, right?
Hmm?
The Actinomyces are good bugs.
They're good bugs. Okay.
Able to attach to.
Oh, well, when does the average first tooth appear in a baby?
6 months.
Yes, 6 months. I've got my first grandchild. I'm going crazy about his teeth, right? He now has 3 teeth. I can't be happier. Beautiful little pearly teeth, right? And he's now about 8.5 months old. First bud appeared when he was about 5.5 months. He's absolutely normal. Now, what is interesting is that the antibodies to this bug don't appear because the teeth aren't there, right? Even the good bugs that bind to salivary pellicle, you're not going to see antibodies to them either. Strep sanguinus, Actinomyces viscosus, these are organisms that really bind to pellicle exceedingly well. You're not going to see antibody titers against these bugs until the teeth erupt and there's been enough time for mother-to-child transmission of the karyogenic organisms. Now you've got dentate children. The tooth eruption brings a second wave of strep organisms. A second wave. And here you see strep sanguinus and strep mutans. The problem is that strep mutans is a highly effective organism that produces tremendous amounts of lactic acid during the fermentation process. Strep sanguinus doesn't approach that at all. But strep sanguinus binds exceedingly well to salivary pellicle. So that's a good bug. And again, strep mutans does not bind well to salivary pellicle. Now, when do you start seeing antibodies against strep mutans? When the baby's about 1 year old. Okay? And these antibodies will identify specific components of strep mutans. We'll talk about serotype-specific carbohydrate molecules, where, remember that serotype C is the one that is most important of the mutant streptococci, of which strep mutans belongs. Serotype C is the one that plays a major role in caries development. These guys, protein 1/2, this is a term used for adhesin molecules. These are adhesins. So these are molecules that will bind poorly, but will bind to salivary pellicle. And glucosyl transferase. This is an enzyme. See, it says ACE at the end of it. ACE. That means we got an enzyme here. So glucosyltransferase is an enzyme that will cleave sucrose and make chains of glucose. And those chains of glucose become glucans. That's the sticky stuff on the surface of the organism. Anticoic acids. So those are agents that are inflammatory. But we're not going to talk about them in regards to strep mutans. Certain streps play a role in soft tissue inflammation, but we're not going to be talking about that. But the antibodies against these agents can be seen by the time the baby is about 1 year old. And by 10 years of age, the secretory IgA levels in this child are about comparable to the adult levels. And that's what you see, that's the range. Okay? Stop me if you think if you're not understanding something or if you want me to repeat anything. This is just a cartoon of secretory IgA. It's a dimer. So when Dr. Levine talked to you about antibodies, he talked to you about the FAB part and the FC part? Yes?
Yes.
So the FAB part is the variable part. That's the part that identifies an antigen or an epitope in an antigen. That's unique to that antibody. And the FC part, which is over here, sticks out. So let's say this is the antibody, the FC part sticks out. That's usually used for opsonization in the context of phagocytosis. There are FC receptors on the surface of PMNs. But notice, this is a dimer, and there's this thing called the J chain that connects these two dimers. So this is not a very good opsonizing antibody. But what it does have, it has the capacity of binding to 2 sites. Maybe more than 2 sites, because each one of these can bind. This can bind to 4 sites. So it's effective in binding to bacteria. And then there's this thing called the secretory component, which is made inside the cell, and I'll show you how. That secretory component is real important to promote stability of the molecule and to prevent or to reduce the ability of other organisms to chop it up. So there are enzymes produced by bacteria in order to combat secretory IgA. All right, so this is the molecule of interest, a dimer with a J chain binding the 2 FC parts and a secretory component that protects it to a great degree from bacterial enzymatic disruption. All right, I always like to talk about the history of stuff and how they came to realize the importance of this molecule. This group, in 1975, demonstrated or showed that low titers of parotid salivary gland produced secretory IgA corresponds to an increase in dental caries. And that was associated with studies relating IgA deficiency and susceptibility to caries. So they identified that this IgA deficiency afflicts 1 in 1,000 people, and this is the kind of stuff that you'll see in children who suffer from this. So when they put these kids to sleep, they inadvertently use bottle feeding, and bottle feeding has sugar in it, so they exacerbate the problem even more. And so they get rampant caries beyond belief. Not only are you adding stuff to generate caries, but you're adding it in a child where there is tremendous deficiency in the important molecule that combats it. Okay? So the secretory IgA against strep mutans found a lot through the parotid gland, meaning it's generated in the parotid gland. We'll talk about that in a couple minutes. It produces, and these secretory IgA molecules can recognize the major serotypes of strep mutans. Now remember, the serotypes are based on the carbohydrate moieties on the surface of the bug. So there are multiple different serotypes for strep mutans. And this guy, George Hashishingalis, he was then a graduate student or a postdoc where he started his work in the area of strep mutans, determined that parotid-derived secretory IgA against these surface antigens that are the adhesins, if you generate secretory IgA molecules against them, which means you've isolated them away from the bug, and you now immunize an animal and you generate antibodies against it. If you use those antibodies, it blocks the ability of strep mutans to adhere to hydroxyapatite, which is kind of like enamel that is coated with saliva. So this is an in vitro experiment. This was the first attempt in '92 to demonstrate that secretory IgA against a particular virulence factor can block the ability of strep mutans, in this case, to adhere, which suggests that there's a mechanism of protection. So let's assume you inject strep mutans near the submandibular gland or near the parotid gland. All right? Plasma cells with antibodies that will pump out, plasma cells that will pump out secretory IgA, or both monomeric and dimeric IgA molecules against all kinds of different antigens from the organism that you injected, will be produced. Now most of this, over 2/3, are dimers. There is a small component of monomers that are found in the blood that are opsonizing. Okay, but that is a minority. These guys are the majority. What they do is they bind to receptors on the basal surface of the epithelium of the acini in the gland. Once they bind to this, it's taken in vacuoles, and the receptor here intercalates with this particular molecule, the dimer, and that becomes the secretory component. And then it's released into the lumen. Another way to look at it is this way. There's your receptor, here's your dimeric IgA, and this becomes that secretory component. And to belabor it even further, you've got this. These are B cells located in the submucosa. They convert to plasma cells that produce the dimeric immunoglobulin A, bind to this receptor, internalized, and the receptor becomes the secretory component. Another way to look at it, more histologically, is this is the structure of the acinus, intercalated duct, striated duct, and in all of these places, the plasma cells will generate these IgA molecules, which will go in and become the mature secretory IgA molecules, released into the lumen. Now, the major salivary glands, submandibular, sublingual, parotid, they produce about 70%, if not more, of total salivary secretory IgA. 30%, maybe a little less, comes from the minor salivary glands. They're all doing it the same way, what I described before. 3 times as much IgA is produced per day in a human than IgG. Much more secretory IgA is produced, or IgA is produced in an individual than IgG. Just look at some of this. The big place is in the small intestine. Look how much is being produced when compared to IgG. In saliva, although it's smaller, it's like 100 times more. But IgG is released into the saliva. It's of no effect IgG needs the tissue, right? It's an opsonizing antibody. It's got a free Fc component. That free Fc component, I said to you, is important in phagocytosis. But the secretory IgA molecule is important for binding to free bacteria in order to reduce the load of those bacteria that are being produced nonstop in the gut and in the oral cavity. Primary function is to prevent microbial adherence. So in saliva, it's a great molecule, and we have to recognize its importance. What else did I say? Oh yeah. This is very interesting. And maybe it's a defense mechanism for a good bug to prevent or to reduce the ability of secretory IgA to adhere to the good bug and remove it. So it's been found that Strep sanguinus produces IgA-specific proteases that attack that secretory component and destabilize the secretory IgA. But this is a bug we want to be binding to the teeth. So this is an interesting evolutionary development of how a good bug overcomes the mechanism of protection that we have against bad bugs. Needless to say, there are certain periodontal pathogens like PG, Porphyromonas gingivalis, that can do that as well. All right? And we'll talk about some of those organisms next time because I've got 2 more sessions with you. Unfortunately, 1 hour tomorrow and 1 hour the next day where we have to introduce periodontal, the story around periodontal disease. Okay, so specific immunity against dental caries. Caries are correlated with a high increase of secretory IgA titers. So this is very interesting. I told you before that caries are associated, or the incidence of caries, or individuals who don't have or can't produce titers of secretory IgA are at high risk for caries, right? And now I'm saying caries are correlated with a high titers of secretory IgA. Well, that's in response to those organisms. So the body is generating high titers of secretory IgA. And that's what you should be seeing. Okay? So this isn't conflicting with what I said before. This is a normal thing that should be happening. You get an agent, a foreign agent like a bacterium. It multiplies, increases in your body. You're expecting to get adaptive immunity to kick in, and in this case, secretory IgA titers to go up. The big question is, are these antibodies actually protective? Because what I'm talking about is passive immunization, right? There's active hyperimmunization, and there is passive immunization. What do I mean by passive immunization? Yeah?
Do you mean like innate immunity?
No? I'm not talking about the innate immune system. I'm talking about the fact that we are passively introduced to organisms that come into us. And in response to that, the immune system generates a certain level, a certain titer of antibodies, right? A certain titer of antibodies. That's a normal response to a foreign body, a bug. That's passive. Is that effective? In certain cases, yes. In many cases, no. So that's why if you are actively hyperimmunizing with a particular virulence factor, that's a vaccine, the specificity of that increased titer goes up because those antibodies are now at really high titers against a particular virulence factor. So you anticipate a better response. Now, add to that and this was found primarily in the periodontal immunology research arena, not all antibodies produced by people are created equal. Think about this. The FAB part of the antibody is the specific part, right? And the FC part, like I said before, is the constant part. So it's the FAB part that provides efficacy, if you will, or specificity, right? So the affinity of the FAB part of the molecule to bind to a particular epitope of an antigen is dependent upon how well it recognizes that particular epitope. That's called affinity. You can have antibodies with really high affinity. Then there's another aspect to this dynamic, and it's called avidity. Avidity is a situation where the affinity may be strong, but the retention of the complex may be weak. So it may be binding and coming off, binding and coming off, binding and coming off, right? So if it comes off, it's not effective. So there are studies being conducted in people. The University of Washington, there was this famous guy, periodontist, PhD guy I worked with. His name was Roy Page. Roy's now retired. He was a proponent of the specificity of antibodies generated by different people in response to treatment. Have you done scaling and root planing yet? On each other? You have? Yeah, not everyone's done it. Just well, all right. Wait until you start really scaling each other. That's going to be great. You dig deep into the, into the tissue, and you start causing bleeding to happen, right? Now, what do you think that does? When you do a deep cleaning of an individual because he's got a lot of plaque, accumulated plaque for years, it's hard, it's difficult to remove. What do you think you're doing when you're doing that sort of stuff?
Mechanically disrupting the biofilm?
You're absolutely disrupting the biofilm. Some people believe you're also, with a lot of aggressiveness, because the removal of the plaque sometimes is very difficult, you're also removing some of the tooth material, right? You're scraping not just that, but you're also scraping tooth material. So there are these conservatives who believe that overdoing scaling root planing can be detrimental. But I'm not going there. What I'm talking about is the fact that you're causing bleeding. Now, when you're causing bleeding, what do you think in regards to what I'm talking about is happening?
Introducing bacteria to the bloodstream.
Yes, you're introducing bacteria to the bloodstream. And when you introduce bacteria to the bloodstream, what do you think happens? Antibodies get generated. Now all of a sudden the titer spikes. So that is a good thing. When a periodontist or a general dentist does that sort of thing, it accelerates and increases antibody titers. So what Roy Page at the University of Washington was able to determine is that there's interpersonal variability in the quality of the antibodies being produced to that activity. And where is that quality difference occurring? In the FAB part of the molecule. Some are really good at identifying the epitopes, right? Some are not so good at identifying the epitopes. Some are really good at identifying the epitopes and maintaining the complex so that you get phagocytosis. Others are really good at identifying the epitope, but their avidity is bad, so that the complex breaks down and you don't get good phagocytosis. That's primarily in the context of periodontal disease. We'll talk about that tomorrow. But In this case, a similar thing may be occurring. The antibodies generated against the cariogenic bacteria and those virulence factors may not be qualitatively effective in eradicating the organism. And remember, secretory IgA is a great antibody to adhere to bacteria in solution, meaning in the saliva. Okay? Now this is another busy slide that talks about naturally induced immunity, NII. That's passive immunity. That was what I was talking about before. You get caries. The natural event that happens is an increase in titer. But those titers are very general, and if they do increase, they're not really increasing well. They may not be protective. And then I talk about hyperimmunization, which is actual vaccination. Results in elevation of the antibodies to therapeutic and preventive levels. That's been shown over and over and over again. And the aim of the vaccine is to reduce the number of pathogens and or to interfere with metabolic activity. If you can block strep mutans' ability to produce lactic acid, and I'll show you how you can do that without vaccination, a thing called fluoride. Fluoride has been shown to do exactly that. That's another reason why I'm not a strong proponent for vaccines in regards to this particular disease. The criteria for effective hyperimmunization: identify the bad guy. Find out who the bad guy is. Identify the best target in that bad guy. Is it glucosyltransferase? Is it the adhesin? Is it glucans? Okay. Identify which component of the immune system should be targeted. In this case, secretory IgA. Is there evidence that hyperimmunization will work? Once you've done it, you're going to have to do extensive work in animals in order to determine whether your targeted virulence factor, where you generate hyperimmunized a situation where the secretory IgA titer goes really high, is eventually effective in reducing caries. Okay, I'm going to stop. We'll take a break, and hopefully we'll finish the session by 10 to 12. All right? And if you want, because it's, I think, in the afternoon, do you have anything after the class? I think we're 3 to 4. Is that right? If you want, we can spend a few minutes afterwards. All right? Okay. Thursday. Is that 3 to 4?
Yes.
Yeah, that's what I meant.
That works as well, so it hasn't been announced yet, but we'll have a review session from the professor that evening, provided as a host as well.
Okay.
Thursday, so.
All right, I just won't be able to attend.
That's okay, no worries.
Yeah, I have to submit my questions by Friday, because I'm going to overseas. So, yeah, I'll make certain that I don't have anything after at 4, and we'll spend some time together. All right, criteria for cariogenicity. What are the criteria that we use to identify if an organism is cariogenic? It has to exhibit tropism for teeth. Fancy word for predilection, desire, i.e. binding to, it needs that substrate, the tooth surface. It must be acidogenic. Okay, it's got to generate acid, in this case lactic acid, that can be destructive to the tooth surface or to the tooth in general. It has to be aciduric. What does aciduric mean? Huh? It has to endure acid. It has to be able to thrive in a low pH environment. And it has to be able to utilize refined sugar, in this case sucrose. The caveman, the ancient man, who was a carnivore, never had a problem with caries. The problem with ancient man is that he wore down the teeth, right? A lot. Maybe he was a bruxus, he'd brux, and he'd grind. But the teeth were intact. More modern man started to find wheat and started to generate bread and carbs, things we love, right? We can't live without. And now all of a sudden we get caries. Now all of these things, the common denominator is sugar. So those are the 4 criteria established by Ernest Neubrun, probably the most noted cariologist in the 20th century from UCSF. He had a fantastic cariology course that I listened in on back when I visited him at UCSF. Tremendous individual. All right, lactic acid bacteria as prime suspects. So this is a heterogeneous family of bacteria. It's not just one bacteria. It's a whole gamish of different streptococcal organisms. And I'm not talking about lactobacilli here. Now, some are good and some are bad. I can't emphasize that more. The strep sanguinuses are examples of good bacteria. The streptomyces, which don't have a tropism to teeth, they have a tropism to mucoidal surfaces, those are seen as good bacteria. The strep mutans, the strep sobrinuses, those are bad bacteria. The common denominator is that they all ferment sugars and form lactic acid as an end product. Okay, so these cariogenic organisms have these properties. Interestingly enough, lactic acid is less volatile. It has the ability to stay put for lengthy periods of time. It can chelate calcium and obviously facilitates demineralization of the enamel. Now, this is an important point. They all form these extracellular glucose polymers called glucans. They all have the ability to generate this network of strands of glucose or strands of fructose outside of their domain that other organisms readily bind to. And they do this through the activation of GTF, or glucosyltransferase. Now there is something called fructosyltransferase, which will cleave sucrose and generate chains of fructose. And there is glucosyltransferase, which will do the same thing for glucose. Okay? This is not that important. I don't know why I did that. But you can see here that there are some organisms that can protect the oral environment from worse pathogens. Those are the strep sanguinuses of the world, right? And some others. The aspect of the bad part of it is that they can generate enamel caries. Lactobacilli that I mentioned to you, in difference to streptococci, which are cocci, these are rods, these are bacilli, but they're both gram-positives. And they both appear in chains. They both can protect the oral environment from worse pathogens. Okay, not just in the oral cavity, but other orifices of the body. And they promote dentinal caries. So the difference between this and this has to promote enamel caries, and then these guys come in, they're highly acidotic. Lactobacilli love to thrive in very low pH. And these guys are really good at pickling things. I don't know why I put that there. But for food. You like pickles? They will generate really good pickles. Okay, Embden-Meyerhoff pathway. What do you need to know about this? You need to know that glucose undergoes a process by which at the end of the day lactic acid is produced. Some streps do this extremely well and very efficiently, and a lot of lactic acid is produced. Others don't do it as efficiently. But very importantly, this is really, really important, fluoride can inhibit number 9, called enolase, phosphopyruvate hydratase, which will convert this thing into pyruvic acid. So you will get the inhibition of this particular enzyme before it becomes lactic acid. So fluoride can effectively reduce lactic acid production in these organisms. Very important take-home message. All right? Very, very, very important take-home message. Okay, what's important about this slide? The family, or the umbrella term, is mutant streptococci. Now, how did it get that name, mutans? From mutate. Because when they first discovered it, there's a common denominator or a common phenotype when you look at these organisms on a plate. You shine a light on it, and look, they look glassy. They look like they're mutating. That, you know, microbiologists have a weird way of calling things, and they see it, and then they describe it, and they own it. So we have to live with it. Mutant streptococci. A group of streptococcal species most closely associated with caries of smooth surfaces, pits, and fissures. Okay? I'll describe a particular study done in the 70s at the University of Alabama in rats, where they demonstrated for the first time really effective ways of eliminating smooth surface caries, but not pit and fissure caries. There are 6 serotypes of mutant streptococci. Now remember, mutant streptococci is the umbrella name. And there are these different streptococcal species within the mutant streptococci that are associated with different animals. Not just humans, but these other animals, and this is far from the complete list. There are other animals that can generate caries in their teeth because of different specific streptococcal cariogenic bacteria that are indigenous to those animals. So strep mutans and strep sabrinus that I mentioned to you before, these are the different serotypes, C, E, and F for strep mutans, D, G, and H for strep sabrinus. Okay? C, serotype C is the predominant group associated with enamel surfaces. Greater than 80% of children who present with caries present with serotype C, strep mutans. All right? Now these guys are found in rats, monkeys, hamsters. There's another one in rats. These are great animals to study because they have indigenous forms of cariogenic bacteria similar to strep mutans. Okay, this is the study done by Michalik et al. And presented in 1976. And what they did was they intentionally took a group of rats and they injected strep mutans, whole-killed strep mutans. So the antigens were still there, the bacteria were no longer thriving, and they injected them near the submandibular gland. They found that essentially what they did was they hyperimmunized those rats. They fed them a cariogenic diet, and they compared them to animals that were not immunized. Okay? And they looked at the outcome of caries in the mouths of these rats. So this is baseline. They used a means caries score. It has to do with the level of penetration of the lesion. Okay? And so these were not immunized, not infected. This is baseline. Now, if you look at the buccal surfaces or the proximal surfaces of the not immunized and infected, they got a pretty deep caries in the buccal and in the proximal areas. But if you looked at the immunized and infected, so these were the animals that were intentionally immunized with strep mutans and fed a cariogenic diet, the buccal level of caries was closer to baseline. And the proximal level of caries was closer to baseline, which means there's protection. However, the pit and fissure areas were not well protected. That's a take-home message. The pit and fissure areas of the teeth are extremely susceptible to caries. And that's why you learn to do sealants, right? You've done the sealants on little kids, right? There's a reason why you do sealants on the pit and fissures. Did anyone explain this to you guys as to why you're doing pit and fissure sealants this way? They did. Did they bring out the McKinley study? Okay. They don't complete the story. Okay, so results suggest that protection is at best location dependent. Sulca protection requires additional protection through sealants. Okay, virulence factors of strep mutans. Now this is not a very good diagram of what I wanted to convey to you, but it'll have to do. We're going to talk about certain virulence factors that are associated with sucrose uptake. We're going to talk about the antigen 1,2 that binds to anchoring molecules. We're going to talk about glucan production. We're going to talk about glucan binding proteins, that other bacteria bind to the glucan generated by a bacterium that's already bound to the tube. And we're also going to talk about something that's not here, called dextranase. I'll mention dextranase when we get to it, because it's an extremely important example of a strategy that strep mutans uses when it feels that the mother has already gotten to the understanding that she's got to take sucrose out of the diet of her child. Now all of a sudden, the organism is starving. Now, the organism that's starving is still virulent. That's a very, very important take-home message. All right, we'll talk about that. Okay. This is not that important. I use this for other purposes. But we're going to talk about glucosyl transferases, the adhesins, the dextranase, the glucan binding protein, and maybe some surface polymers. So what happens initially? I told you that strep mutans is not an organism that readily binds to salivary pellicle. But you only need one, right? You only need one bug to bind somewhere on a tooth surface in a child's mouth to be dangerous. And that's what happens. Now that bug is bound. And it uses these adhesins, which you'll see in the literature referred to as SA1-2 or B or P1 for strep mutans, or surface protein antigen A for strep subrinus. They play the same role. They will bind to things like PRPs. Remember PRPs, proline-rich peptides, or staphyrin? Those were the anchoring molecules in the salivary pellicle. Although their binding affinity is greater than 1,000-fold less than strep salivarius. But they will bind. Once they bind, and there is sugar in the milieu, they will start generating this enzyme called glucosyl transferase. Okay? And when that happens, it'll start breaking up the sucrose into chains of glucose, and the other enzyme, fructosyltransferase, will do the same thing but generate chains of fructose. And what you will begin to see is, here's an example of a mutant streptococcus. That's a general term for a karyogenic organism. That there's a pellicle glycoprotein. This could be a proline-rich peptide, for example. It will bind to that. You'll have, and this is a serotype carbohydrate. Now, some people have shown that glucosyltransferase, when it's produced, will bind to the serotype, to the surface of the carbohydrate antigen. Now it's on the surface. It'll take a molecule of sucrose, it'll break it up, and it will start producing glucans. So these glucans, this is a representation of these glucans. Yeah, this is the representation of the glucans. Will become exceedingly sticky, a tremendous branching of these molecules out from the surface of the bug. Another strep will come in. It's got glucan binding proteins on it, and it will bind to the glucan that's being generated by the first bug. Now these guys are going to do the same thing, and they're going to have other bugs binding to them that are going to do the same thing, and now you're going to generate a community of biofilm that's highly karyogenic. So now this is important. Water-insoluble glucan versus water-soluble glucan. It's an important concept. Now how do you get to the water-insoluble versus soluble? Well, this is called dextran. And dextran is a glucan made up of alpha-1,6 linkages. That's an example of water-soluble glucan. Okay? That's not a very strong glucan. Okay? On the other hand, the water-insoluble glucan is made up of alpha-1,3 linkages. Water insoluble. So dextran is rich in alpha-1,6 linkages, and the water insoluble glucan called mutan is rich in alpha-1,3 linkages. You got to worry more about the alpha-1,3 linkages because they're the ones that keep the bug really strongly adherent to the tube. However, the alpha-1,6 linkages, remember that, because I'm going to come back to it. There's a strategy that strep mutans uses in the event that it feels starved of the substrate, where it uses the alpha-1,6 linkages to break up that glucan in order to store energy inside of it. I'll talk about that in a minute. So, to repeat, this is the sticky stuff that these karyogenic bacteria use to adhere to one another as a strategy to circumvent the poor adherent ability of the organisms to bind to salivary pellicle. These are homopolymers of glucose. They're also homopolymers of fructose. They feature gazillion branches. I can't emphasize how many branches. Okay? And there are two types. The alpha-1,6 dextran, the alpha-1,3 mutan. The alpha-1,6 is water soluble, the alpha-1,3 is water insoluble. And there are studies that demonstrate that if you generate antibodies that impede GTF function, you can protect the animals. Again, if you're going to generate, if you want to generate a vaccine, this is a very, very good target to interfere with or to inhibit.
Is there ever a glucan that alternates with the 1,6 and 1,3s in the same molecule?
Yes, I just showed you that.
Oh, those are
This is,
different ones.
you've got the branches coming off of this. Sometimes they're 1,6 and the branches coming off are at 1,3, or vice versa. Or they're or they're linked, you know what I mean?
So it won't be like.
But the 1,3s, if they're homo 1,3s, those are the ones that you have to worry about in terms of binding, all right? If you have something like this, and let's say the bug uses, I'll already talk about it, dextranase. Dextranase knows to cleave the alpha-1,6. If it's cleaving the alpha-1,6 to which alpha-1,3 is bound, it's going to destroy the adherent capacity. But if it's homopolymers alpha-1,3 only, dextranase won't work on it. All right? And so the bug will still be bound well. Do you remember this? Did I ever show you this before? Okay. And remember I showed you this palisading, intermittent dark light, dark light, dark light, dark light, dark light kind of interaction, or whatever, staining pattern. And remember I showed you this down here. Those are the fusobacterial organisms. This is from an experiment done in 1972 at the University of UCLA with dental students where EPON strips were put on the buccal surfaces of premolars and molars, and the kids were asked not to brush for a couple of days. And then they took those strips off and they processed them and they stained them, and they found this kind of organizational community forming on the EPON strips. All of these, to a great degree, are cocci and maybe bacilli, but gram-positive. All of a sudden, you're starting to see this. This is now introducing those bridging organisms that go from gingivitis to periodontitis. But this kind of stuff, they didn't understand why it's stained that way. Now we know. This area is made up of glucans that are water soluble. And this area is made up of glucans that are water insoluble. All right? So that's a take-home message. And what's the function of glucan? Well, obviously, it promotes plaque accumulation. It permits the organism that binds poorly to the salivary pellicle to outwit and circumvent that feature, and therefore you're getting plaque formation in organisms that bind poorly to the tooth to begin with. They're molecular sieves. They permit water retention. They're a secondary attachment apparatus for bugs, and especially for bugs that don't bind well, primarily to the salivary pellicle. They strengthen the attachment of producing organism to the tooth, and it enables producing organisms to control their microenvironment. And again, antibodies against the glucans have been shown to be protective. The adhesins, a little bit on the adhesins. There's a lot of these surface molecules on strep mutans and on strep sabrinus. As I mentioned before, these are the names given in the literature for those adhesins that bind to anchoring molecules, and importantly, bind poorly to anchoring molecules. This is the name for it in strep sabrinus. A lot of it, as I said, 35% of all surface proteins are made up of these adhesins. They adhere to tooth in the absence of sucrose, but they adhere poorly. And I don't want to go into this, but obviously if they lack this, they're not going to bind well to, not at all, to experimental pellicles. And the rest isn't all that important. Here is dextranase. Now this is a very, very important take-home message. What dextranase likes to do, and it's expressed in those cariogenic organisms when they sense that sucrose, that substrate, is no longer available to continue generating the glucans. Okay? because remember, they ferment sugars for energy. What happens is dextranase then gets expressed, and it specifically breaks down polymers of glucose in the alpha-1,6 linkage. So they modify the glucan product of the glucosyltransferrin. The glucan product changes, and it becomes more of an alpha-1,3. But what happens in response to the breakdown? It converts this into an alpha-1,4 linkage. And that's internalized as glycogen. So now you have internalized alpha-1,4 linked glucose inside the cell of an organism to prevent its death. Okay? It uses it as storage for energy purposes when sucrose is no longer available. It continues to generate lactic acid. That's an extremely important take-home message. And then clearly if you mutate and now the dextranase doesn't work, or if you mutate the adhesin molecules, these organisms are avirulent. That's not surprising. So the take-home message with this, for you guys, as clinicians, is that a child comes to you and you're scolding the parent because the child has rampant caries. And you're educating the parent and telling the parent that feeding processed sugar to the child is dangerous. And this is why the child has rampant caries. So you do what you do really well to reconstruct that mouth, to remove all the caries, and 6 months later, the mom comes back with the child for a checkup. You turn to the mom and you said, "Mrs. Jones, I'm discovering new carious lesions, new cavities, 6 months after I completely removed and treated the teeth for caries. What happened?" And the mom tells you emphatically that she listened to you, and the moment they got home, she threw out all the candy bars, no Coca-Cola, no Pepsi, none of that. And yet, the child comes back and now has generated more caries. And the reason is that these organisms are really smart. They sense that sucrose is no longer available, and they generate these dextranases. And they cleave the alpha-1,6, they convert it to alpha-1,4, they store it as glycogen, and they continue to ferment the glycogen and generate lactic acid. That's an extremely important take-home message. So what you have to do is not just remove the substrate, you got to remove the bug. And there are ways to do that. First of all, there's some products out of the Scandinavian countries who are really good at this, where you can take a dipstick, it's kind of like a, pedodontists do it, where they want to know what the titer, or the level of strep mutans per ml of saliva is in the mouth of a child. And you can determine that using a particular product that they have that you put on the tongue of the tooth and you put it into media overnight. And there is a table that they provide you with that you can compare the number of colonies formed on that stick with a standard. And that'll tell you how many colonies per ml there are. And I can't remember what the number, 5 times 10 to the 5 per ml. Anything above that, I think, is dangerous. And anything below that is what you strive for. So you as the clinician have to make every effort to reduce the bacterial load in the mouth of the child to less than 5 times 10 to the 5th per ml of saliva. Okay? That's a really important point that I want to convey to you. Okay. I think we're going to finish early. Serotype defining carbohydrate antigens. I told you there are like 8 different serotypes, and serotype C is the one that's most important for caries by strep mutans in the US. And these are the ones for strep sabrinus. And what's interesting is that studies have shown that these guys don't just play a role as being a serotype, so you can identify them in individuals. The carbohydrate antigen appears to play a role in being the pedestal on which the glucosyltransferase sits in order to promote the production of these glucans. And so people have suggested that these carbohydrate antigens, these serotype-defining carbohydrate antigens, are proposed targets for Caries vaccine because antibodies against these carbohydrate antigens, if you bind them specifically to these antigens, they will be protective and prevent GTF from being able to promote the production of glucans. That's an important take-home message. What number slide is this? 34? I'm finished. You get to go to lunch early. So if you have any questions, we can start a review now if you want. Want to do a review now? A little bit? All right, so obviously we haven't talked about perio, so we'll start doing that tomorrow. What do I want you to know? In innate immunity, I focus a lot, if you recall, on toll-like receptors, on pattern recognition receptors, pathogen-associated molecular patterns. What are they? Why are they important? What's the outcome of the interaction between PAMPs and PRRs? Why is that important in inflammation? Now, complement I didn't touch on. I'd like to think that Dr. Levine talked about complement, right? Tomorrow, I'm going to focus on dysbiosis. That's a really, in perio, that is a hot topic, where organisms shift in their community and certain keystone pathogens, as they're called, emerge. And these keystone pathogens are able to subvert the local immune response. One way they do it is to affect complement. So I'm hopeful that you will have had enough introduction into complement so that I can use your knowledge and talk about dysbiosis and not have to talk about complement. Okay? I'm hopeful that you guys will remember what Dr. Levine taught you. So salivary innate immune functions, obviously that's key. Saliva's filled with all kinds of innate response elements. And the ones that I touched upon, mucins, there are primarily 2 types. Which one does what? Which one does the other thing? Why are they important? What are they made up of? How do they function? What is the salivary pellicle? What are the anchoring molecules? What's unique about proline-rich peptides? How does it function? What are histatins? What's the role of histatins in the oral cavity? What else? Lysozyme, lactoferrin, obviously, I mentioned those. The antimicrobial peptides, which are primarily produced by the epithelium, what are they? How do they function? I focus primarily on LL37 and beta defensins. How do they work? I think that's pretty much it. Here, obviously, the centrality of secretory IgA, the virulence factors of strep mutans, the importance of dextranase in a clinical setting, I guess that's it. I can't think about anything else. Now, tomorrow, I'll be focusing on the issue of dysbiosis with certain bacteria as keystone pathogens. I'll be giving you examples of how this dysbiosis occurs with diagrams. I'll define what a keystone pathogen is. I'll demonstrate how the commensal bacteria shift so that a keystone pathogen promotes what is referred to as a community of pathobionts, which are organisms that thrive in the shifted community that leads to more inflammation. I'll give you examples of what these keystone pathogens do in subverting the immune response, how they affect neutrophils, how they affect epithelial cells, how they affect cytokines, how they affect complement, how do they affect macrophages, how they affect T cells. And that's stuff that I'm going to want you to be able to know and remember. And that's going to be tomorrow. And then on Thursday, I'm going to focus on neutrophils because they are an extremely important cell in the context of periodontal disease. I'm going to redo some of my presentations. The dysbiosis story is really hot, so I'm going to be emphasizing that. So you're going to get, to a great degree, a pretty fresh, new presentation. So I'm going to work on that today. And I'm going to give it to Heather either, depending on if I get it done today or early tomorrow. When do we meet tomorrow? At 10? 10, right? Yeah, so I'll get it to you guys before we meet. All right? So then, when I finish with perio, we'll have a quick chat on the importance of... You had a question?
Yeah, is the oral mucosal strategy...
Can you raise your voice?
Is the oral mucosal
Coastal strategy.
Is the main idea from that just that there's 3 levels?
Right.
Not so much the specific, or...
Right, so I try and simplify these complex ideas so that you have an understanding of the dynamic in the oral cavity. So that's why I broke it down into the fluid phase, what I call the passive phase, or the what? Static phase. It's static only because it's a barrier. But it's not static in terms of the response to a challenge, right? And you got to know what that challenge is and how the epithelium responds to that challenge. And then there is the recruitment phase, and that recruitment phase is caused by that barrier that I call the static phase, because it promotes the release of chemokines that come in to recruit cells from the bloodstream, right? I'm trying to do that in order to give you an understanding of how the oral cavity responds to challenges. All right? So that's what that's about. Anything else?
Is GTF...
Glucosyl transfers. Yeah, so you have time to ask me questions about the material tomorrow and Thursday. All right? Thank you.
Thank you.